Alright guys, in this video we're going to talk about the electron transport chain. In order to understand this, you must understand the steps that took place prior to this. And that was glycolysis. In glycolysis, glucose enters into the cell and is broken up into two molecules of pyruvate. In that process, ATP is made, two net ATPs, and also you make two molecules of NADH. Okay? And that's going to be very important in this process. Next, the pyruvate is processed, and one of the carbons is removed from it in the form of carbon dioxide, and acetyl-CoA connects to it and enters the matrix of the mitochondria. In this case, you make a couple of things. You make some more NADH, you make a little bit of ATP, you make another electron transporter by the name of FADH2, and then this cycle continues to go on as long as oxygen is present. However, oxygen is not needed for this process to take place. Like, it's not in the process, but this process does not take place unless oxygen is present. So it's important to understand that. Now, the electron transport chain takes place inside the inner membrane of the mitochondria. And in this inner membrane, you have a series of proteins known as complexes. I'm going to draw these guys real quick. So in this picture that I'm drawing here, I'm going to label these guys just a second. But we have a couple of things that are very important. The first things to mention about this is that I'm going to call this the intermembrane space. And this right here is going to be the matrix. This last protein that I'm drawing here is actually an ATP synthase. I want to talk about it later. It's responsible for making ATP. So let's go look at these guys real quick. These proteins are numbered using Roman numerals, in which this is complex 1, this is complex 2, this is complex 3, this is complex 4, and this right here is an ATP synthase. And an ATP synthase is responsible for actually making ATP. Well, let's go start the story off real quick. The first thing is NADH. Remember, NADH was NAD and is reduced to NADH. What NADH does is that it gives its electron to complex 1, or it reduces it. It becomes NAD+, the electron comes through. Okay? Now, what's important about this electron is that once this electron comes through, is that there's some hydrogen ions that are present in the matrix. People always ask why. Well, because they're around the cell, period, but because a lot of chemical process to take place. When this electron comes through, an hydrogen ion is pushed from the matrix and into the intermembrane space. This electron then is taken up from complex 1 to this complex right here. I'm going to label it Q. The real name of this is ubiquinone. Ubiquinone is an electron transporter. It takes the electron from complex 1 to complex 2. Once that electron is at complex 2, the same process happens, in which a hydrogen ion shoots through and goes through the other side. Let's go make sure it's the same color. The other issue with this is that there's another molecule that's present, FADH2. And FADH2 takes its electrons and it gives it to complex 2, not to complex 1. Now, complex 2 does not shoot electrons, but what it does is that ubiquinone also takes its electron and sends it here to complex 3. So there's really a coupling, two electrons together in that case. So as a result, we get more hydrogens that are shot up through the matrix. There's another molecule here, and this molecule, the name of it is known as cytochrome C. If you're going to take a biochemistry class in college, you'll know more about cytochrome C than you want to learn about. But cytochrome C takes the electrons and then transports it to complex 4. Once that happens, you probably guessed, an electron is shot there, but then a hydrogen ion or a proton is shot up. Now, the issue at the end of this is that now I have electrons that are in this last complex, complex 4. And they have no other space. That's the end of the road. That's the end of the electron transport chain. And that's where our good friend oxygen comes into play. The purpose of oxygen is that oxygen takes the last electron, and in the process, what it does, it makes water, and that's where that last those electrons go. So this is the reason why you breathe. This is the only reason why you need air, is because what oxygen does is that it's an electron acceptor. Okay, if you have no oxygen, that means that the electron is stuck in complex 4. And since this is like an assembly line, that means that it can't go on. It stops everything. It halts it right there. That's also the problem with cyanide, is that cyanide attaches to complex 4, changes the configuration of it, and as a result also halts the process. Okay? Now, here's the big story here. The big story here is that as this process is taking place, you start getting an influx of pluses inside the intermembrane space, because they're being pumped into this space. And inside the matrix, it becomes predominantly negative. So in a sense, this thing becomes polarized, like a battery. The membrane becomes polarized. There's a difference in charge on each side of the membrane. Well, what happens at this point is that these positives that are up here, they want to get back down, but they can't, because they can't go to the membrane. If they try to go to the membrane, they just bounce off and have to stay inside this side. And this is where this ATP synthase comes into play. The ATP synthase provides a way for protons to go from the positive end back to the negative side. Remember, they're attracted because opposite charges attract to one another. They use this gradient, they use this change in charge to power the ATP synthase. And it makes the ATP synthase literally spins around, and as it does that, it takes ADP, adenosine diphosphate, plus inorganic phosphate, and it makes ATP in the process. So this ATP synthase makes this, but its power source is this influctuation of gradient that's taken place that was because of these protein complexes. So it's using that to make ATP, and it makes the most ATP that we've seen yet. Okay, for each glucose molecule, for one glucose, you make 34 ATPs just from this process. In order to have it, you must have NADH, you must have FADH2, you must have oxygen present. And obviously the membrane proteins are important. So with this, look at this review, go back through it, understand the context of what's happening and taking place, and understand the final product, the thing that you really need here, is ATP. So it's a complex process. However, it's fairly simple. The entire purpose of it is to make a small battery, a small potential gradient in between the intermembrane space and the matrix that's down here in order to power the ATP synthase. Another name that you can see for this as well is phosphorylation. And I believe inside of your sheet that I use is oxidative phosphorylation. So if you see that oxidative phosphorylation, that's basically the process of the electron transport chain and producing ATP as a result of it.